Alright class, so this is another representation of the neuromuscular junction. On the very top, we do see the axon terminal or the terminal bouton of a motor neuron, and we see the synaptic vesicles filled with acetylcholine. We see the synaptic cleft. We also see the motor end plate with ligand-gated sodium channels. Now the main point, we also see the T-tubules and we see the sarcoplasmic reticulum. This is sarcoplasmic reticulum, and we do see that calcium is in the sarcoplasmic reticulum. Now the main concept or the main thing that I want you to understand here is that these green arrows, these are action potentials coming down sweeping across the sarcolemma, down the T-tubules. And as this action potential comes down the T-tubules, remember, the voltage sensitive receptors or the dihydropyridine receptors, they undergo conformational changes resulting in opening of the calcium release channels, and calcium then is released. So this is action potential, and in response to that, calcium is released into the sarcoplasm. Do you see all the blue balls? These are calcium floating in the sarcoplasm. Calcium will go and bind to troponin protein, and as calcium binds to troponin, the tropomyosin complex changes shape, and that opens up the active site of actin proteins. We will see all this in the next upcoming slide. Okay, so we will start at 4 here. We will start here. Now let's label a few things before we start. We see the thin filament, right? We also see the thick filament. Now these blue balls, these are actin proteins. The rope-like protein is called tropomyosin, and the clip-like yellow proteins, these ones, these are troponin. To which one does calcium bind? You were right, calcium binds to troponin. Alright, these are the myosin molecules as you see. So this is a myosin molecule. And we know that the myosin molecule will bind to actin, and as there is binding or attachment of myosin to actin, the thick and the thin, this will help the thick and the thin filaments to slide past each other, resulting in sarcomere to contract. As individual sarcomere contracts, the entire myofibril contracts. When many myofibrils contract, the muscle fiber contracts, and when many muscle fibers contract, the fascicle contract, and when many fascicles contract, the muscle contract. Okay, all right, so now let's start with calcium. So we have calcium now in the sarcoplasm. So calcium comes and binds to the troponin. So this is calcium binding to troponin. As calcium binds to troponin, the tropomyosin changes shape, or there is conformational change in tropomyosin, which is a rope-like protein. And as that happens, the troponin-tropomyosin complex is removed, exposing the active sites on the actin proteins. So these are the active sites which are now exposed. Do you see that? Therefore, I repeat, calcium comes and binds to troponin, which are the yellow proteins here. In response to that, the tropomyosin changes shape, and the troponin-tropomyosin complex is now removed, exposing the active sites on the actin proteins. Now let's focus on the myosin molecule. The myosin molecule is in cocked position and it has ADP, an inorganic phosphate or PI, attached to it. This is high energy or cocked state of myosin. The idea is myosin is ready. It is ready to attach to the active site of actin. Okay, but it could not do so because the troponin and the tropomyosin was covering the active sites, so it could not attach. Now that calcium has come into play and calcium caused this conformational change of tropomyosin, leaving the active sites open, the myosin will now swing in and attach. So this is attachment. If you look here, we see that the myosin heads have attached to the active site of the actin molecules. This is attachment. As the myosin heads attach, okay, so this is attachment. Immediately after it attaches, the myosin heads flex. Do you see that the head is flexing? So there is flexion of the myosin heads, and as there is flexion, the inorganic phosphate or the PI, that gets released. So the flexion of the myosin heads releases the inorganic phosphate or PI. Now as the myosin heads flex, that pulls the thin filaments closer towards the M line. Remember the sarcomere? This is the M line. So as the myosin heads flex, the thin filaments are brought closer to the M line. That results in shortening of the sarcomere. Okay. Now, the myosin head obviously pulls on the actin filament, and now we have sliding towards the M line. And after sliding, ADP is released. So now at the end we have ADP being released. So after flexion, we have something like this. We have the myosin still attached to the actin. However, we do not have ADP, we do not have PI attached to the myosin now. Now at this point, the myosin wants to detach. This is detachment. But it needs help of an ATP to detach. Therefore, an ATP comes in. We have the ATP here. That comes in and helps the myosin head to detach. Remember, ATP helps to detach. And after the myosin heads detach, the myosin heads will go back to its cocked position or high energy position. And this is called recocking. This stage is recocking. Now the ATP gets hydrolyzed to help the myosin heads to come back to its high energy cocked position. And the cycle gets repeated as long as we have free calcium available in the sarcoplasm. So let's do it one more time. We start here again, and in this segment we start at number 4, and here we see calcium coming in and binding to troponin, which are yellow colored proteins. Binding of calcium to troponin causes conformational change or shape change in tropomyosin, and the troponin-tropomyosin complex is removed, exposing the active sites on the actin proteins. The myosin molecule is in high energy state with ADP and inorganic phosphate, denoted by PI. So the myosin heads have ADP and PI attached to it, and it's ready. It's in high energy state. It's ready for an attachment. In the next step, there is attachment where the myosin head will swing to attach to the active sites on actin proteins. And after attachment, the myosin heads will flex by a process called flexion. And as the myosin heads flex, that pulls the thin filaments towards the M line, resulting in shortening of the sarcomere. Flexion of myosin heads causes release of inorganic phosphate or PI. And after the M line is pulled, after the thin filament is pulled towards the M line, at the end, ADP is released. So now we have a flexed myosin head attached to actin with no ADP, no PI attached to it. Therefore, for detachment of the myosin head from the active site of actin, we require an ATP. An ATP comes into play, and ATP helps the myosin heads to detach from the active sites of actin. Therefore, the myosin heads detach, and in the next step, going back to number 4, in the next step, the myosin heads come back to its original cocked position or high energy position by ATP hydrolysis. So the ATP that helped the myosin heads to detach will now get hydrolyzed to bring the myosin head back to its cocked position. And this is how the actin and myosin proteins interact with each other, resulting in sliding of the thick and thin filaments, and the sliding of the thick and thin filaments ultimately result in contraction of the sarcomere. I'll stop here and then start with a new video.